So I'm requesting all the participants, please mute your mics so that there will be no echo or noise.
Okay, so let's start our topic. Today I would like to discuss experimental approaches to treatment of neurodegenerative disorders, namely Parkinson's and Alzheimer's disease, with a focus on modulation of proteostasis and neuroinflammation. So first of all, why this topic is so acute and actual today? Well, because dementia is among the priority of health problems of the World Health Organization Mental Health Gap Program due to the increasing of lifespan and the percentage of elderly people in the society all over the world. And due to this reason, the percentage of diseases related to aging is also increasing, and neurodegenerative disorders are among the most severe and hardly to cope with. So World Health Organization estimates that there are currently more than 35 million people with dementia all over the world, and they forecast that this number will double by 2030 and more than triple by 2050. Dementia has a significant social and economic impact on society, as you may know, since those patients are disabled, and it takes many years to take care of them with no hope to any improvement, because till now all the neurodegenerative disorders are incurable. Alzheimer's disease is the most common cause of dementia at the old age. Around 60 to 70% of all cases are for this disorder. So old age is the main risk factor for the development of Alzheimer's disease, and every 5 years after the age of 65, this risk increases about 2 times. And the second most common neurodegenerative disorder is Parkinson's disease. According to current estimations, the more than 6 million people suffer from this disorder all over the world. So what we know about these disorders and why it is so hard to find treatment to these diseases. In the case of, for instance, some infectious disease like so popular COVID-19 in these days, you know exactly the reason, the pathogen, virus or bacteria, and you may study this pathogen and find the exact pathological mechanisms, and there are a lot of successful strategies to prevent or cure this type of disorders. And in case of neurodegenerative disorders, unfortunately, we don't have successful strategies still. As I mentioned, most, maybe all the neurodegenerative disorders till now are incurable. And what is even worse, we don't know the exact mechanism or why these disorders happen, and we still are in search for these mechanisms and uh, hope to treatment. So what we know for sure that neurodegenerative disorders are linked to neuronal loss in certain brain areas. This is cerebral cortex and subcortical areas in Alzheimer's disease, and mainly dopaminergic structures like substantia nigra and uh, striatum in case of Parkinson's disease. So first method that was applied to cope with these disorders, of course, was symptomatic due to the loss of neurons of certain um, and certain neurotransmitters. This deficit of neurotransmission was tried to substitute uh, uh, by, for instance, application of acetylcholine esterase inhibitors to increase the level of acetylcholine in Alzheimer's disease, or L-DOPA to substitute dopaminergic deficit in PD. Of course, those symptomatic approaches do not halt the progression of the disease, don't prevent the certain deficits, and are not effective actually. So therefore, now major efforts are aimed at the discovery of a new effective pathogenesis relevant therapy for these disorders that would stop the disease progression and ameliorate most of the disturbed functions. So what we know more about these disorders? Both Alzheimer's disease and Parkinson's disease are closely related to disturbances in the protein metabolism, and are related to the disturbances in metabolism of certain proteins. In case of Alzheimer's disease, this is amyloid, and at the late stages, tau protein. So around 20 years, a little bit more ago, the amyloid cascade hypothesis was formed. And in the slide, you may see its main points. So both for the genetic forms that are not actually very common, about only 1 to 5%, and the rest forms are sporadic, non-genetic. So in both cases, the main reason and the initiation of this cascade is related to the accumulation and oligomerization of amyloid beta. And at the first stages, the oligomers of amyloid beta uh, cause synaptic dysfunction. Then the situation is worsened, and microglia and astrocytic inflammation, neuroinflammation occurs. Uh, Actually, this hub connected to microglia was supported by recent findings. The importance of this uh, hub to the pathogenesis of Alzheimer's diseases. Just recently published paper in the beginning of this year confirmed that risk of Alzheimer's disease is related to certain genes of microglia response to amyloid beta. Then neuronal ionic homeostasis is compromised, and oxidative stress and mitochondria dysfunction occurred. It's also accompanied with altered activities of certain enzymes like kinases and phosphatases, and that led to formation of hyperphosphorylated tau and neuro and tangles inside the neurons consisted of this hyperphosphorylated tau. And all this led to the massive synaptic dysfunction and neuronal loss. So the similar processes occur in Parkinson's disease too, but in this case, the protein is alpha-synuclein, and the pathological accumulation led its pathological accumulation leads to the formation of Lewy bodies inside the neurons that also produces neuronal loss in the certain brain structures. So, taking into account these findings and results, the approaches were shifted to targeting specific protein, amyloid or alpha-synuclein, or specific enzymes of their production. Actually, the first approach was not successful due to the functional role of these proteins in neurons and the central nervous system. So good amyloid and alpha-synuclein take part in many important processes in neurons, like neuronal plasticity and neurotransmitter metabolism. So concerning the second approach, production of A beta is associated with intracellular cleavage of the amyloid precursor protein, APP. Its amyloidogenic cleavage pathway is associated with the enzyme beta secretase. So many researchers study the possibility of simulating APP cleavage along with a non-amylogenic pathway, activating alpha secretase or suppressing amylogenic transformation of the peptide by the beta and gamma secretase. But significant success in this direction was not obtained, first of all, due to the profound side effects of these drugs and insufficient influence on the restoration of cognitive function. And besides side effects, we should note that most of these drugs and clinical trials with these drugs were applied at this stage of moderate and severe AD, when actually they wouldn't be effective due to the appearance of all the other pathological mechanisms like microglia activation, neuroinflammation, uh, severe metabolic defects, synaptic loss, and uh, at the severe stage of Alzheimer's disease, there is a massive loss of neurons, and in this case, you may expect any success only in combination with some strong regenerative technologies, maybe like uh, cell technologies, pluripotent cells, um, applying of neurotrophic factors, and so on. So, of course, it's better to uh, apply any intervention at the early stages of the disorder and to apply multi-target approach, since neurodegenerative disorders has a multifactorial etiology and involve various pathological processes in addition to neurotoxicity of protein aggregates, as I have already mentioned, that often closely interact and overlap. So one of the modern trends is multi-purpose or multi-target therapy, including drug combinations aimed at various important pathogenetic hubs in the course of neurodegenerative disorders, is currently regarded as relevant and promising approach. There are certain good examples of applying of this approach now. One of these is GV971. It is now at the third stage of clinical trial in China, and according to the reports, it has quite good effect in people with mild to moderate Alzheimer's disease. It's oligosaccharide derived from marine organisms that affect such pathogenic hubs of AD development, like inhibition of A-beta fibril formation, neuroinflammation, and reconditioning of dysbiosis in gut microbiota. And now I would like to share some of recent results from my lab. that are related to our today's topic. As I mentioned, one of the problem of drug development, a big problem of drug development, is side effects and toxicity of potential drugs. The one of the trend in the pharmacy is drug repositioning or drug reprofiling. This is the process of redeveloping a compound for application in different pathology and finding new therapeutic indications for the existing drugs. The premise of this strategy is that the drugs that have previously passed clinical trials will minimize the risk of failure in future late-stage clinical trials due to toxicity and thus led to faster drug approvals. With our colleagues from Taiwan, we had a series of studies with ceftriaxone. Ceftriaxone is a well-known antibiotic drug and it is widely used to cope with bacterial infections of wide spectrum. What is important for potential application as psychotropic drug, it is highly soluble in water and it penetrates blood-brain barrier. Recently, the property of ceftriaxone to enhance the activity of glutamate transporter, GLT1, and thus normalize levels of glutamate hyperactivity and reduce excitotoxicity was discovered. That led to the research of ceftriaxone for possible repositioning for neuroprotection. Increased excitotoxicity appeared to play an important role in the mechanism of development of neurodegenerative disorders and cognitive dysfunction in animal models of Alzheimer's and Parkinson's disease. So we hypothesized the beneficial effect of this drug for treatment of cognitive deficits in Alzheimer's and PD-like pathology. And indeed, using this drug, we found that it restored cognitive function and neuronal deficits in the models of Alzheimer's disease. We used two models, pharmacological model in mice induced by amyloid beta administration into lateral ventricles, and genetic model of accelerated senescence and sporadic Alzheimer's disease produced with selective breeding, that is Oxys rat strain. So we found beneficial effects of ceftriaxone on behavior. It totally restored the compromised learning function and episodic memory in Oxys rats, and also it improved neuronal deficits that was estimated by the density of neurons in the hippocampus. And similar effects were observed in the model of PD-like pathology induced by the stereotaxic selective dopaminergic neurotoxin MPTP into substantia nigra in rats. So we also found the restoration of working spatial memory novel object recognition, and these beneficial behavioral effects were accompanied by the restoration of density and activity of neurons in the hippocampus. Moreover, we found new targets and mechanism of neuroprotective effects of ceftriaxone. That is modulation of gene expression levels of enzymes involved into the metabolism of amyloid beta in the brain. This gene is related to A beta production, and this gene are encoding proteins related to A beta degradation. We also found the decrease of A beta accumulation in the frontal cortex of A beta-like mice, AD-like mice, and also ceftriaxone reduced microglia activation in the frontal cortex. So our studies gave new insights into mechanism of ceftriaxone, of its neuroprotective action, and it's really proved that this drug has multi-target effects. So it is a promising pharmacological tool for prevention of cognitive decline at AD and PD-like pathology. And I should mention that our Taiwanese colleagues started clinical trial with ceftriaxone in PD patients with cognitive impairments, and it's now ongoing study in Taiwan. So I would also like to mention that Professor Singh and me and two more of our colleagues from Taiwan and France applied to the frontiers in neuroscience a proposal to edit an article collection related to this important topic. Here you may see the potential title of this research topic. I hope it will be launched in near future, and we will have an opportunity to review this topic in more details. And take home message. So I would like to say that neurodegenerative disorders have a multifactorial etiology and involve various pathological processes in addition to neurotoxicity of protein aggregates that often closely interact and overlap. So multi-purpose or multi-target therapy including drug combinations aimed at various important pathogenetic hubs in the course of these neurodegenerative disorders is regarded currently as a relevant and promising approach. And of course, when you choose these combinations, you should take into account the stage of the disease to choose to select appropriate treatment approaches and combinations. And here I thank you for your attention, and if you have any questions, I would be happy to answer them. Oh, Professor Singh, I think you should turn off your microphone.
Yeah, can you hear?
Yeah.
Yeah. So thank you very much, Professor Tikhonova, for such a nice presentation. And now I would like to request all the audience, if they have any questions, then they can ask to Professor Tikhonova. Actually, here is the, light has gone and storm is there. So it's a bit dark here. So if anybody have any question, then please, one by one. No one have any question? If you have any question, then you can type or you can raise your hand one by one. And the topic, Professor Tikhonova was just explaining that we are just editing a special issue in the Frontiers of Neurosciences that is totally based on the presentation aspects, that we need to find out the suitable therapeutic potential for the Alzheimer's and Parkinson's disease. So in coming future, that issue will be launched. So you all, if you all are working on this, if you are working on AD or PD or both aspects, then you can submit the articles there in the Frontiers. And so I think nobody have any question. So
I'm glad that everything was clear.
yeah, I hope so, because nobody is asking any question. So I think now you can stop your screen sharing, so that we can,
Okay.
yeah.
Okay.
Now all the participants are here in one screen. So if at the last, I would like to thank Professor Tikhonova and all the participants who have given me an opportunity to present this webinar in front of you all. And Dr. Birla.
I think that there are some questions.
Oh, is there? Yeah.
How to prevent
Sachin,
PD?
Sachin is asking, is there any side effects of the citraxone? And somebody else to ask. You can unmute your mic and you can ask the question. No need to type. If anybody wants to ask a question, then unmute your mic and you can ask. Dr. Dharmendra, Dr. Novi and Budi, please unmute your mic and ask the question to Professor Tikhonova. Novian, please ask your question.
Thank you. How to prevent Parkinson's diseases?
Okay, so thank you so much for your question and your attention. Actually, there is still no effective treatment.
His question is also include, Professor Tikhonova, sorry for interruption. His question is also include, can we improve this with the exercise or yoga? Is there any reports?
Yeah, I understand. Yes, actually, there are some findings on Alzheimer's disease with relation to physical exercise and maybe yoga. That this approach may improve the course of, or maybe postpone the development of Alzheimer's disease. Unfortunately, with Parkinson's disease, I haven't read such data or results.
Okay. So anybody have any, Sachin, I think Sachin was wanted to, any side effect of, you can ask Sachin, you can unmute your mic and you can ask the question. Sachin, can you hear me? Please unmute your mic and you can ask the question. Okay, I think he's shying in speaking. So okay, he typed
No, no
something.
problem.
Any side effects of the ciprofloxacin? Thank you for this. Any side effects? His question is regarding side effects of this compound.
Well, there were actually a clinical trial with lateral amyotrophic sclerosis with this drug, and there were some side effects on biliary system, hepatic system. That's it. So I will ask my Taiwanese collaborators about their clinical trial in Parkinson's disease, but I think that they are using lower doses of ceftriaxone, and maybe there are less side effects at these doses.
Okay, I hope Sachin is okay with this answer. Birla is asking some question. Birla, you can speak. You can unmute your mic. Your voice is not coming. So his question is the microglia response to alpha-synuclein like A beta and AD?
Sorry?
His question is there any microglial response to alpha-synuclein in A beta Alzheimer's disease?
Yes, in animal models, this effect was proved many times that microglia responses to A beta, and it led to its activation, hyperactivation, and transformation to bad microglia that destroys synaptic contacts and produces a lot of pro-inflammatory cytokines that have bad effects on neurons. And just recently, the paper I mentioned, they confirmed it with genetic research in human.
Okay, anybody else have any question?
I'm sorry, I'm Dr. Gandhi.
Yeah, hello, sir.
I'm sorry I missed that because I got the mail late, but certainly I would request that, I mean, if I can get some sort of a copy of the presentation, maybe.
Sure, yes.
I was at AIMS for about 20 years, from 70 to 90.
Mm-hmm.
And then I was retired from Department of Biotechnology, Ministry of Science and Technology, Government of India. Mm-hmm. That was in 2006. Mm-hmm. I am 74, and I have recently, I've seen that last 2-3 years, I'm having some sort of essential tumor, these tremors.
Okay.
So it was of interest to me. So I would like to see, how, I can participate in the studies, or how can I support the program? I can help the program.
Nice. So it will be honor to have you like Professor Tikhonova. He's a very senior person and he has a lot of experience even. So yeah, we can collaborate and we can do a lot of things. So what is your take on his question or his query, whatever he's asking? So have you, sir, have you any question, specific question regarding his talk? Because I think you missed some portion of his talk. You joined late.
I'm sorry, I missed a major portion of that. When I could get in, you know, after I received the mail.
Yeah.
Almost he was concluding it.
Oh, so sorry.
I'm so sorry for my own mistake.
No, it's all right. So somebody is joining still. Amar Pratap. He is still joining in the talk. So I think I will allow him. So, okay. Any other question from any other participants? Please, quickly, if I have any question, then please ask. Because we are running out of time. It's for already past 10 minutes past. So if you will not have any question, then I will share the, people will find this recording of this presentation on the YouTube channel of the ISET Foundation. So at the end, I would like to tell something about this foundation. The ISET Foundation is basically, it's a scientific foundation, and Professor Tikhonova is the advisory board member of that foundation too. So this foundation is basically working in the field of science and technology promotion in the rural area of India, and also at the same time, sorry, Saleh, have any question? Yes, please.
Yes, Christopher. I just want to have a question, please.
Yeah, please.
Okay. When the patient should consider about the surgery of the Parkinson, when's the exactly time for the considering the operation or surgery?
Professor Tikhonova, have you got the question?
Yeah. As far as I understood, the question was about the best time for interventions or treatment. Actually, as we can see, the process aggravates with time. And so at early stages, the interventions, the treatment would be, of course, more effective because you would address the less harms of these pathological processes. And at the late stages, I think it's a very great challenge to improve all the disturbed functions, including the massive loss of neurons. So it's the matter of early diagnostics, and this direction is also actively developed nowadays to diagnose neurodegenerative disorders at the early stages, when we can prevent their progression and restore the function. And actually, it is considered that there are around 20 years prior the manifestation of symptoms when the Alzheimer or Parkinson disease actually starts. The first signs, with the first processes like appearance of oligomers and some synaptic dysfunction. So I hope the success in the early diagnostics will markedly improve the treatment.
Okay. So, Saleh, are you clear with the answer? Are you satisfied?
Yes, sir. I'm satisfied now for the.
Okay. Now Mohit have the next question. He raised that. Mohit, please ask your question. Please unmute your mic and you can ask the question. Mohit Kumar. Mohit, can you hear me? Please ask the question. Okay, I think is not getting you or any problem is there. So at the end, I would like to thank Professor Tikhonova for her valuable time. And I'd like to thanks to all the participants for giving such valuable time for their busy schedule. And I'd like to thanks Dr. Briz, who has joined a bit late, but yeah, I will share the recording of this presentation of this Professor Tikhonova through YouTube channel of the foundation, so you can find over there. And maybe next time I'd be delighted to have you in another, because we have a next session of the next speaker, which is which will talk about the career development, like Professor Manu Bora from USA. So next talk will be on 20th morning. So hope that will you all, if you will be interested, then you can join over there. And so at the end, I would like to thank Professor Tikhonova again. And thank you so much. Yeah, thank you so much to all of you for joining. Yeah, it's honor for me. Yeah. Okay, thank you. Bye to all of you. Yeah.
Very much.
Okay, bye-bye.
Bye-bye.